Hey everyone, in this video we are going to cover the difference between genome sequencing and SNP genotyping. And we're going to learn a little bit about how SNP genotyping is performed. So essentially genome sequencing, and we talked a little bit about genome sequencing I think in video 2101. Now genome sequencing is pretty great, but it's pretty expensive too. So let's take chromosome here, and no matter how many chromosomes there are in a genome, and let's take a little segment of that right here, and I'll diagram it right here. And one of the great things about genome sequencing is, we can determine the identity of every base along this piece of DNA. So every single one, we can determine the identity. We can determine the entire sequence. So it doesn't matter whether it's an SNV, a known SNV, or a known SNP. Doesn't matter if we had no previous information of what the sequence was. We can determine every single position. So the problem with genome sequencing, though, it's still rather expensive. So I think it's about maybe $2,000 to maybe determine the sequence of a human genome, and it might be even more to get a perfect sequence, but you could probably get most of it for $2,000. So scientists who are doing like giant studies that need lots of data and lots of genome information from a large number of people, well, what they can do is SNP genotyping. And there are companies that provide SNP genotyping and can provide you a lot of information based on your SNP genotypes, your SNP profile, and they can provide you ancestry information and some health information. And we'll take a look at the health component of how they can provide you health information in the next video. But here we're going to talk about the process of how SNP genotyping works. So essentially, we talked about I just talked about how genome sequencing can give you the entire sequence of a genome, so every base at every position. So with SNP genotyping, well, you already need to know where the SNPs are in the genome. And remember from the last video, SNPs are single nucleotide variants that are rather common in the population. So to be an SNP, the SNV, to be considered an SNP, an SNV needs to be found in more than 1% of the population. So they're rather common. 1% doesn't seem like that much, but with 8 billion people, 1%, yeah, that's pretty high. So now let's assume these are all SNPs. And let's assume also that these are about 250,000 base pairs apart. So 2.5 mega base pairs too. So 250,000 base pairs apart. So each of these 2.5 mega base pairs apart. So obviously this is not to scale. And also in the previous video we talked about each human genome on average has about an SNP. Well, they're found about every 300 base pairs. So obviously if we just detect these SNPs, we're not detecting every SNP in this part of the genome. Now let's say this SNP can be a G or an A. We also talked about how SNPs in the human population tend to be found in two versions, a major allele and a minor allele. So we normally don't detect 4 different alleles for each SNP, even though there are 4 bases. Usually a major allele and a minor allele. So let's assume with each of these, I hope you can see this, but this is a G allele and A allele. And the first one I put, this is the major allele. The second one is the minor allele. For each of these SNPs are represented by a major allele and a minor allele. And let's say we know this is an individual here, and we do some SNP genotyping. We determine there's a G here and a C here, and let's say there's the major allele at every one. Now, also in the previous video, we talked about how if we find a G here, based on analysis of other sequences, we can usually predict the sequences that are going to be found here, because these SNPs tend to be inherited as haplotype blocks. So if we know the identities of SNPs all the way along this chromosome, we could probably predict with pretty good accuracy the sequences of the SNPs and also the sequences between the SNPs. Well, I should rephrase that. So we could probably predict the identity of the SNPs between these based on the fact that SNPs spread through the population as haplotype blocks. And people with the same SNPs are likely to have the same sequences between those SNPs. So even if we don't know exactly what that sequence is in those individuals, if they have the SNPs, same SNPs, they're likely to have the same sequences between those SNPs. So that's just a little bit about SNPs, and I'm going to come back to that when we talk about genome-wide association studies and why they are so powerful at finding the genetic causes of complex diseases. But here we're mostly going to focus on how do we determine what these SNP genotypes, what the SNP genotype is at each of these SNPs. And the technology that's most commonly used can detect up to 1 million SNP genotypes. So that's a lot, right? We talked about how the average genome is only, different by 5 million sites, I think, relative to the reference genome. So 1 million SNPs, that's like able to detect 1/5 of the genetic variability in a human, right? Or between humans. So that's a lot of SNPs. So let's take a look at how this genotyping process works. So essentially, so SNP genotyping. Now, a company that might performs SNP genotyping services is 23andMe Incorporated. Maybe you heard about them. Ancestry.com, and there are probably some others out there. In the technology they use, they don't use their own technology. They tend to use technology from a company called Illumina. And the technology is great. So I'm actually a 23andMe customer. I think maybe 2011 or 2012, I subscribed to the service, and now my parents have, my daughters have, and my wife has. So it's pretty neat. And I think my brothers use Ancestry too, my brother and uncle. So both of these are pretty good. I'm sure they're all very similar. So essentially, let's talk about how this process works. So let's say this is an individual, just a person, John Doe. Let's call him John Doe. So what he'll do is John Doe here will spit into a tube, and spit enough that he will put 2 milliliters of saliva, 2 milliliters of saliva into the tube. Then he will cap the tube, and the tube in the cap is a little bit of, I think, an ethanol solution or something that stabilizes the saliva. So when he caps it, the cap kind of pierces the cap and releases all of that stabilization solution mixes with the saliva and seals the tube tight, and drops it in the mail and sends it off to the company, whether it be 23andMe or Ancestry.com. And what they do is they will harvest genomic DNA from the cells I guess the cheek cells or tongue cells or whatever, the small amount of cells that are in the saliva. You get genomic DNA from the cells. They will use PCR, which we will talk about in the next video, next lecture, sorry. PCR to amplify all of the genomic DNA because there's not quite enough genomic DNA in those cells to perform this genotyping. So what they do is they use PCR to amplify all the genomic DNA, so making many copies of all the genomic DNA in the sample, and then they break the DNA or shear it, shear DNA to small pieces, and then they denature the DNA, and by now we know what denature means, right? Making all the double-stranded pieces single-stranded. And so let's say all these pieces are, let's say they're about 100 or 200 bases long. So the whole genome is represented by all these single-stranded pieces of DNA. And each part of the genome is, you know, present in many, many copies. Let's say 1,000 copies of the genome. Each piece of the genome is represented by 1,000 little fragments. Okay, so then we have all these little pieces of DNA. And the next step is to take all those pieces of DNA and put them on this microscope slide-shaped bead chip. Microscope slide-shaped or sized bead chip. Okay, so it looks like a microscope slide. And on this slide are going to be 24 chambers. And I tried to put 12 here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I'll put another one down here. Make the slide a little longer. So, and why not? I'll put the others in here. And each one of these chambers is for genotyping a different person. So John's DNA sample here, John Doe's DNA sample, we're going to put that right on there. Okay, then we could put 23 other samples over there. So we could genotype, you know, 24 people for 1 million SNPs each with this technology. Okay, so how does that work? Okay, so in this little chamber right here, there are 1 million microscopic beads that are sort of etched into the chamber, or embedded into the chamber, embedded into the surface of the chamber. And each bead detects genotypes a different SNP. So each bead genotypes a different SNP. Okay, so how does that work? So let's look at one bead. So imagine this is the surface of the slide, and this is the bead that is sort of embedded. This is one of the million beads that is embedded into the surface there. Now that bead is going to have attached to it what we are going to call an SNP detecting oligonucleotide. Now an oligonucleotide is just a single strand of DNA. They're usually kind of short, between 20 and 50 nucleotides. And here, let's just, I don't know the length exactly, but let's say they're 50 nucleotides long. And I'm not going to diagram it to scale, though. You'll see in the next video, I think in the next diagram, I think I'm going to actually put a sequence on there. So I'm not going to put all 50 nucleotides. So each bead will have 1,000 identical SNP detecting oligonucleotides. Oh yeah, I want to call these SDOs. SNP detecting oligonucleotides. So the 5' ends, so is a DNA. It's a DNA strand, so there's a 5' end and a 3' end. So this is the 5' end, and the 5' ends of these 1,000 identical oligonucleotides are attached to the bead. And the 3' ends, which end with an OH group, right? 3' OH group, those are all free. And if this is a solution of water or buffer or something over these beads, you know, these will be floating in the solution, attached to the beads at the 5' ends. Okay, that's one bead there, and okay, there'll be another bead over here, and another bead over here. Each one has a different SDO. Each one has 1,000 SDOs, but each SDO has a different sequence. And each sequence is designed to genotype a different SNP. So let's say this one detects SNP1, this one detects the genotype of SNP2, this one detects the genotype of SNP3. Okay, so what does the sequence look like? Well, let's take a look here. Okay, so here I have genotype of John Doe at SNP1. Let's say John Doe has 2 Gs at this SNP1. Now, we wouldn't need to genotype John's SNPs if we already knew what they are, but just in order for me to explain how the process works, let's assume, you know, okay, this is John's genotype. So this is the SNP right here, G, and John is diploid, so he has 2 alleles for this SNP, one here and one here. So it's homozygous for this allele, for this SNP. So how's the genotyping process work? Well, those SDOs, the sequence of the SDO is going to be right here. I'm going to draw a box around it. Remember the SDO is single-stranded? So essentially it's going to be identical to this single strand right here. So ACC, ACAC, GA, GGT. It'll start with an A, ends with a T. If it was only, what, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, if it was only 12 nucleotides long, it's probably going to be longer than that, right? But for this example, let's assume it's only 12 nucleotides long. It's going to end right here at that T. So I can diagram that right here. So let me see if I can keep this in frame here, or in the whatever whatever you call that thing with a camera when it's pointed on something, in the view of the camera. I guess that's what we'll say. Okay, so let's see, A, C, C, A, C, A, C, G, A, G, G, T. Oh, perfect, perfect. Okay, and there's I need my eraser here. Okay, so that's the SDO, the 5' end down here. Remember, there are 1,000 of these. There are 1,000 of these identical molecules stuck to this bead. And maybe you also remember, okay, so what happened? We added the DNA. Let me see if I can find that sheet again. I always throw it on the ground. Oh, here it is. Okay, remember, we shared John's DNA, and then we put it onto the chamber. Now that DNA is floating around here in a buffer, and those single strands can now anneal. They can now anneal to the SDOs. And so we already said John's DNA looks something like this, right? Okay, so we can put TGG. So this little piece of DNA eventually is going to find this, and it's going to anneal to it. And keep in mind, every part of John's genome is, let's say there are 1,000 copies of it. So there are 1,000 of these sequences here. This strand right here, 1,000 of them floating around in the solution above the bead. And eventually they will find it. And so we're not going to end at the A here, right? We're going to keep going. So the products were sheared randomly, so who knows where the DNA will end. But at some point it was sheared and we just have a 5' end there. And it's single-stranded. Now down here, this T right here, well, we don't know what the sequence is, so I'm just going to put ends. Okay, so that's a piece of DNA that was floating around in the solution right here. That's a piece of one of these DNAs right here was from this part of John's genome, or this part down here. And eventually it found that SDO and annealed. And remember, there are 1,000 of these SDOs here, identical. So the other copies of that part that were floating around here are going to stick to those other SDOs. Okay, so what's the next step? So once you let all these little pieces of John's genome attach to the SDOs, and remember there are 1 million different beads with 1 million, each one with a different oligonucleotide, or different SDO attached to the bead. Let them all anneal, and then next step is to add DNA polymerase and these things called ddNTPs. Now, what are ddNTPs? Okay, so that might be kind of confusing. So let me talk about those for a second. So we know what ddNTPs are. Well, if we don't, let's review it. So those are deoxyribonucleotides. So the N stands for A, G, C, or T. So DNTPs that means it means DATP plus DGTP plus DTTP plus which one am I forgetting? CTP. Deoxyribonucleotide means these are DNA nucleotides containing deoxyribose. We've been talking about this, I think, most of the semester, right? So we'll have 3 phosphate groups. I'm not going to diagram each individual group attached to the 5' carbon. 4' carbon, 3' carbon has an OH, right? Because it's the deoxyribonucleotides. Even for ribonucleotides, there's an OH here. There's an H here. So this is where the deoxyribose comes from. We're missing the OH group at this position. And this is where the base is attached to, right here, A, G, C, or T. So ddNTP, this stands for dideoxyribonucleotide. And it means ddATP stands for ddATP, ddGTP, ddTTP, and ddCTP. And so dideoxy, well, essentially what that means is it's identical to a deoxyribonucleotide, but instead of that OH there's no OH there. It's usually an H. So no OH there. And what does that mean if there's no OH? It means a dideoxyribonucleotide is a chain terminator. Chain terminator. So if a DNA polymerase, let's say, is adding, say this is the template, it's the 5' end, 3' end, and it's adding nucleotides. Each time there's an OH group at the 3' end of this nucleotide, right? Okay, good. It can add another nucleotide. It's an OH, it can add another nucleotide. If there's no OH, it can't add another nucleotide. So the chain has been terminated. Okay, so by adding ddNTPs, I need to go back to, by adding ddNTPs, the DNA polymerase can at most extend this by 1 nucleotide. It can only add 1 nucleotide to the SDO. And so I should also say that ddNTPs are fluorescent. FLU, they're fluorescent ddNTPs. So the DNA polymerase can at most add one fluorescent ddNTP at this position. And it's going to depend on the base, on the annealed, that's opposite this position, right? It's going to depend on the base on the annealed DNA. And what is the base? Well, that's the SNP position, right? Like we said, the SDO ends right before the SNP position. So the base that's going to be added depends on the genotype of the individual that supplies the DNA. The genotype of the individual at that SNP. Okay, so I think, if you're a little lost, hold on. I'm going to show you. I'm going to take you one step further so you can see how this process works. Okay, like I said, those ddNTPs are fluorescent. So fluorescent ddNTPs, NTPs. So, and I don't know why they did this. This is the only thing that kind of drives me crazy about this technology. And maybe they updated it in the last year. I haven't reviewed it in the last year. But I'm putting a fluorescent molecule on all of these. And in some technologies, what they do is so that dideoxy, where we're lacking the oxygen, the hydroxyl group on the 3' carbon, they actually attach the fluorescent molecule to that 3' carbon. But you can also attach it to other places. So, and the thing that drives me crazy is they don't use a different color for each one. It's usually 2 colors. So in this one version of the technology from Illumina, that's used by 23andMe, at least as of last year, the A's and the T's are represented by a red fluorescent molecule, and the G's and the C's are attached to both green fluorescent molecules. Okay, but it can still work. This technology still works, even with just 2 colors. It just needs to be designed in a special way. Okay, so here, let's say this is the surface, a small surface area of the slide in the chamber used to genotype John Doe's DNA. And let's look at, let's see, 1, 2, 3. Let's look at 20 different, we won't really look at 20, but here's a small section showing 20 different microscopic beads. So you can imagine we are really zoomed in here to see these. Let's say this bead right here is for detecting SNP1. So the SDOs are designed to detect the genotype of SNP1. These are designed to detect SNP2, 3, 4, 5, so on and so forth. I don't need to label these, but I don't know. Once I get going, I can't stop. Okay. Now, let's say for SNP1, we already know that humans can be either G or A. They can be either a G genotype, they can have a G allele or an A allele. And G is the major allele, and A is the minor allele in the human population. SNP2, humans can be T or C. No other possibilities. So the thousands of humans have been genotyped here. It's always either a T or a C. Now for 3, major allele is a C, minor allele is an A. Okay, so now this slide right here, these B's represent John's DNA. So we added John's sheared single-stranded genomic DNA. It annealed to the SDOs. We added the polymerase. We added the ddNTPs. Okay, now there's red and green everywhere. There's red and green everywhere. If we were to shine a laser on it, we'd see red and green everywhere. So what we need to do is let the polymerase add the ddNTPs, add 1 to each SDO based on John's genotype. And it'll extend the SDOs by 1 position only, because these are ddNTPs. They're chain terminators. And then what we can do is wash off everything. So the only thing stuck to each of these beads now, we can even denature these again. We can raise the temperature and get rid of the annealed DNA. We can wash all that off. So the only thing stuck now are the SDOs that are covalently linked to the beads. And each SDO has been extended by one nucleotide, one fluorescent nucleotide. So next what happens is, using a very powerful microscope attached to an imaging system and a laser, a computer will direct the laser shining on each of these beads, and each bead will emit a certain fluorescent color, either red or green, or a mix of red and green. And that will depend on the ddNTP that was used to extend the oligonucleotides attached to that bead. So let's say for SNP1, the computer with the microscope and the camera and the laser detects a red color. And for SNP2, the computer detects a green color. And for SNP3, computer detects yellow. And then red, green, or yellow for all these too. We don't need to look at those. But I think if you can understand these, you can understand what's going on here. So now, knowing that humans can either be either G or A for SNP1, what can we say? What is John's genotype for SNP1? So we know red is used for A and T, right? So we only see red here. In red, we know he has to be either G or A, and we don't see any green. So we look at the red, we have A is red, G is green. So homozygous A for SNP1. Now we can do the same thing for SNP2. Humans can be either T or C. So let's take a look. T should be red, C should be green. We see SNP2 is green. Okay, so that means John must be homozygous C for SNP2. Now what about SNP3? Humans can be C or A. So C is green, A is red. So we got yellow here. Now when you mix, this is kind of, you don't really know the physics behind this, but if you mix red light and green light, it makes yellow light. If you mix red paint and green paint, it makes, like, I don't know, brown or something. But if you mix light, you know, for some reason it's yellow. So we don't need to understand the physics behind that. We just know if we mix red light and green light, we get yellow. Okay, so what's the genotype then? Well, okay, red is A, green is C, so he must be heterozygous for this SNP. Okay, so the computer can do this with the laser and the microscope, just record all these images and give us John's SNP profile for 1 million, which is essentially genotype, for 1 million SNPs. So pretty cool, huh? This is a really neat technology, and I hope you kind of understand I hope I didn't lose you there. I think it's clearly outlined in the notes too. I think the important part is that the technology only allows for extending each SDO one position. So a fluorescent nucleotide is inserted here, and the nucleotide that's inserted here is dependent on the genotype or the base at that SNP position on the annealed DNA. So pretty neat technology, and once we have this SNP profile, we can do a bunch of things, and we're going to take a look at that in the next video. I think I've been ending a lot of the video saying we're going to take a look at that in the next video, but now I promise next video we're going to take a look at what we can do, or just maybe just one of the things we can do with an SNP profile. But there are lots of things we can do. Okay, so see you in the next video.